alcohol can produce altered motility, producing diarrhea, and also alcohol is associated with a higher risk of colorectal cancer. The alcohol, I have already mentioned that the alcohol is associated with esophageal dysmotility and increased prevalence of GERD and erosive esophagitis. But alcohol on the GI motility, on the gastric motility, in general, the higher concentration of alcohol reduces the GI motility, but lower concentration accelerates the gastric emptying. But acute administration of alcohol inhibit the gastric emptying. So it reduces, and while chronic administration of a large dose of alcohol accelerates the gastric motility. In the small bowel, the consumption by either acute or chronic, it alters the motility of the GI tract, and it can produce malabsorption and can produce hurried peristalsis, diarrhea, and acute administration of alcohol sometimes has been found to inhibit the small bowel transit than producing constipation. But chronic administration of alcohol has been found to accelerate the small bowel transit. So the effect of alcohol is acting on the muscles of the GI tract and alters the motility, and to some extent significantly associated with pathological consequences and disorders. I've already mentioned that GI cancer can be associated with excess alcohol consumption, and the risk is higher when it is associated with the other confounding factors like smoking, or if there is higher metabolic syndromes like obesity. And the most important, the organs affected is esophagus and colorectal cancers, higher incidence of malignancy associated with alcohol. Now, just how the gut defense is altered by alcohol. The alcohol causes alteration of gut microbiota, both composition and the functions of gut microbiome. And so there is a development of leaky gut, so that the gut products like lipopolysaccharides and peptidoglycans enter through the leaky gut into the submucosa and produces inflammatory response. So the intestinal barrier functions is affected by alcohol. Both the transepithelial and the paracellular functions are altered. The alcohol directly can enter the injury of the epithelial injury, the mucosa, and the inflammation produces inflammation. So this is increased leaky gut. And also the tight junction proteins are damaged by the alcohol. So there is paracellular leakage also happens. The relevance of these alcohol-induced gut-derived inflammation occurs within the GI tract and promote the several diseases state like GI cancer and inflammatory bowel disease. Inflammatory bowel disease per se is not directly related to alcohol, but it can worsen the symptoms, particularly if there is active inflammatory bowel disease. Outside the GI tract produces liver disease and neuroinflammations. So what are the modifying factors that the gut-derived inflammation can changes the microbial composition and function, increases the permeability of the intestinal mucosa, and disrupt the immune system of the intestinal intestinal barrier. So disruption of the intestinal microbiota can happen either from the genetic predisposition or biological stress like circadian rhythm disruption and the diet with alcohol. Then it alters the permeability and microbiota composition. So that can lead, this alteration of the gut dysbiosis can be associated with malignancy, some metabolic diseases, GI like inflammatory bowel disease, and food and allergy, celiac disease, liver disease, and neurological problem. So this alcohol effect on immune functions, the alcohol there produces bacterial overgrowth, alter the macrophage function, that there is activation of pro-inflammatory cytokines, produces gut inflammation, and gut inflammations lead to the hepatic parenchymal inflammations, macrophage activity, and this cytokine activation lead to fibrosis. And this gut inflammation, the endotoxins produces systemic inflammation, and also there is associated with neuronal inflammation. So ultimately, this gut-liver axis is the mechanism of hepatic injury due to the alcohol, and these mucosal, the cytokines, the Kupffer cell activity, all these, along with the increased uptake of endotoxin into the blood and the lymph, produces the activation of the inflammatory cascades, and which is responsible for the liver cell injury and organ dysfunctions. So in a one the animal star model, that alcohol produces increased intestinal permeability, and the factor here, it is intestinal hypoxia induced factor 1 alpha activity are expressed, and it is reversed by giving with lactobacillus rhamnosus GG therapy. So the hypoxia induced factor 1 alpha is also important for pathogenesis of hepatic steatosis. And the alcohol is associated with the abundance of these endotoxin producing enterobacteria. And there is reduction in the taxa that produces the short chain fatty acid. These are protective. So short chain fatty acid is reduced, and these particularly the Lagnospiraceae and Ruminococcacea are reduced. So there is alteration of the microbiota by alcohol. So, and also there is alteration in the composition of the bile acid. As the disease associated with with alcohol and with alcoholic liver disease, alcoholic hepatitis and cirrhosis, the total serum bile acid is increased, both conjugated and unconjugated bile acids, but the fecal secondary bile acids and the total fecal bile acids gradually reduced. And these bile acids, and the secretions in the inflammation of activity of in the intestine, all the regulating factors I've discussed, that pro-inflammatory cytokines, increase liposaccharidase, and the reduced activity of FGN90, and this alteration of bile acid is responsible for the inflammation cascade and the linking of the gut-liver axis. And also, when this gut-brain axis is altered, and the alcohol without liver disease can affect the part of the brain of cortex and the extrapyramidal and on the frontal lobe. But when it is affected with alcoholic liver disease, there is further involvement of the parietal lobe, occipital lobe, and also the brainstem. And then these excessive endotoxemia, this ammonia, and the inflammation produces poor brain and cognitive function. And along with this, there is nutritional compromise, which all lead to impaired brain function in the pathogenesis of alcohol-related brain dysfunctions. So this is a summary of all the problems what happen in the process of the progressive disease of the liver. Where the alcoholic liver disease, there is reduction of the protective, that is the some of the bacteria, and also the short chain fatty acids, the fecal secondary bile acids, and reduces, and gradually the microbiota, the viral, fungal, and the bacterial composition are changed. The total bile acids and the fecal primary and secondary bile acid ratio is increased, and the gut permeability is altered, as it is associated with the significant the correlation with the severity of the liver disease. So, and the potentially beneficial changes happen in minimal hepatic encephalopathy when it is treated with symbiotics or prebiotics. There is increased firmicutes, increased lactobacillus, and increased bifidobacterium. But the bacteroides, enterobacteriaceae, they are reduced in patients with cirrhosis, and also in patients with alcoholic without liver disease. The bacteroides is reduced, and the proteobacteria is reduced. The bacilli are high, they are increased. So these are the bad bacteria, and by treating the bacterial composition is altered. So, and there is some changes with the circadian rhythm with the alcohol and gut interaction. There is some progenes, clock genes that has been shown to regulate these lipid transport and motility, and alcohol stimulate these circadian clock proteins, and by knockdown by siRNA, these prevent alcohol-induced permeability. So these are the potential mechanism by which we can modulate the target, the therapy for alcoholic liver disease and alcohol-related damage. These are some of the studies which showed that by altering the gut microbiota in alcoholic liver disease, that from 2008 to 2018, the last two data that is on the FMT, that is fecal microbial transplantation, there is improvement of the in patients, particularly in alcoholic hepatitis. These will be discussed by my other speaker. But just in a nutshell, that these are that some of the animal study and also the human trial, that the modifying the gut microbiota, we can improve the problems of liver disease due to alcohol. So this is to summarize that the effect of alcohol on the GI tract is both direct damage, as I mentioned, and these are mainly due to the acute injury to the GI tract, particularly the esophagus and the stomach, and also chronic damage like can produce diarrhea, malabsorption. But it is more important that indirectly it alters the gut microbiota and mucosal defense. So gut microbiota manipulation, like with probiotics, fecal microbial transplant, and antibiotics, sometimes may be helpful in the treatment of alcohol-related disorders. Thank you. Thank you very much.
It's a wonderful presentation. I think you have made very important, observations in effect of alcohol on GI tract. Maybe if Dr. Neeraj Sharaf is there, I thought I'll ask his comments on this particular talk, because we have a couple of comments that can be made to give value to the topic. Neeraj, could you make some? Is Neeraj there?
So unmute yourself, Neeraj sir.
Yeah, so excellent talk by Dr. Chaudhary. I think he has nicely covered the effect of alcohol on the entire GI tract. And like he said, I think what is important is the direct effects on the mucosa and the effects on, by altering the gut microbiome and its relation to the liver as well. And we have seen that this has implications in management of alcoholic hepatitis as well by changing the gut microbiome. Yeah, so I think a very good talk on this effect of alcohol on the various organs of GI tract.
There is an important lady in our group, Dr. Rupa Banerjee. She would probably comment on, alcohol, GI inflammation, and IBD. I mean, that it's a tripartite relation. Rupa, can you just make your observations?
So she could not able to join that. So she is not in network area.
Fair enough, fair enough. Now I have Dr. Manoj Kumar, who is observing the whole thing. Could you make some comments, sir? Because we, as a panelist, how would you take Sujit's, talk on real-time practice?
Excellent talk. And alcohol as some disease like hypertension, diabetes affects from top to bottom.
Yeah,
so
that's important.
And gut is obviously the main organ which is affected by alcohol. Liver is the main organ, but some changes occur in GI tract also, which leads to all the changes of alcohol manifested disease.
Gurunath, we will go to, if Dr. Dharmesh is on, he wanted to speak, and we request Anil to, just follow. Is Dharmesh ready there?
Yeah, sir, we can go with Dr. Anil first, not a problem, sir.
Oh, great, sir, great. I know you have been always so kind. I don't know how.
Sir, actually,
No.
Some problem was there. It's seems to
No.
be getting sorted out on its own. Courtesy,
No. It's
Sethu sir's blessings.
No. It is you. You people are so teamwork. Anil, I mean, Guru, you can invite.
Sir, since we have 1 more minute, so there is one question from Professor Tayyab. Why do we have variable alcohol-related injury to gut and liver?
So actually, that about 20 to 30% of chronic alcoholic produces liver disease. So that is the main reason that why we are considering that why not all the people are getting affected. So those who are chronic alcoholic, and this gut alteration of the gut defense mechanism is probably the main mechanisms. And I have said that there are a lot of factors that can alter the gut defense. And that is the main mechanism. Maybe that is one of the mechanisms why not all the people who are taking alcohol in the equal extent, only 20 to 30% of the patients are developing liver disease, and only less than that they are developing cirrhosis or alcoholic hepatitis. So
I think I'll dilate on Sujit, but Dharmesh is the correct person to comment. Why only a subgroup of people develop severe liver disease with less of GI tract disease, and there is a group which have severe GI disease and less. I would, whenever you don't know, you blame the genetics. I think that is the one very important.
Yeah,
whenever you don't know, you in the examination you say it is genetic. But there is some very interesting observation. Sometime back, NEJM published a beautiful paper. The difference between women and men. Even generally because of our local cultural factors, our women drink less than men. It will be something close to 8 to 2%, but there is a change. That means if I see 10 cirrhotics, let us say, I mean, most of them would be alcoholic, and then comes other group of diseases. Unless you say you add autoimmune, suddenly the woman might change. But if you take alcohol alone, there is something interesting happening in the stomach, the acetaldehyde mechanism, which is different in men and women. Dharmesh should be the right person to comment.
So it seems that because of the lesser metabolism of alcohol. So alcohol has two first pass. One is the gastric first pass metabolism, and the second one is the liver first pass metabolism. So a proportion of alcohol that is ingested gets metabolized by the gastric ADH, and that seems to be a little deficient in ladies. Yes. And that is the reason why not much of it can be metabolized by the stomach, and it quickly transits into the duodenum. So the gastric emptying for alcohol is very rapid. And when it goes into the duodenum and it then enters into the portal circulation, then the ADH in the hepatic parenchyma for the women is less efficient than men, which means that effectively you have more alcohol in circulation for any amount of alcohol that is ingested between men and women. So that is the reason why they tend to get inebriated earlier. So anybody who gets inebriated earlier will probably put a check on the drinking pattern. So that is the general thinking behind this.
Guru, we can go to the next topic.
Yeah, thank you. For the next session, I would like to invite Dr. Anil Arora from Delhi, and the session is on impact of alcohol on liver, from fatty liver to fibrosis and cirrhosis, and overview. Over to Dr. Anil.
Hello, good evening, everybody. Are my slides visible?
Yes, you are visible.
Okay. I'm thankful to Manthan project, especially headed by Dr. Sethu Babu, for having given me this opportunity. The topic which has been allotted to me is how does alcohol result in sequential damage of liver via fatty liver through alcoholic hepatitis to frank cirrhosis of the liver. WHO, according to the WHO, 5.3% of all deaths which occur in the world occur because of alcohol. Amongst patients who develop alcoholic liver disease, alcoholic hepatitis has most florid presentation. Unfortunately, the treatment is pretty dismal and almost non-existent. Let's see what happens once you imbibe alcohol. Alcohol is quickly absorbed. This is one of the important components of mechanical damage which occurs both in the gut as well as in the liver, that most of the alcohol which we ingest is absorbed through the upper GI tract into the circulation. Only 10% of the unabsorbed alcohol lands up into the colon, And the way it tries to damage the liver and the GI tract is that it has to be ultimately got oxidized. 90% of the alcohol which is ingested is oxidized in the liver. 10% is not only oxidized but is also by non-oxidative metabolic pathways is utilized in the colon. Let's see what happens in the alcohol which comes through the portal vein after absorption through the duodenum and jejunum and stomach into the portal vein. There are three major pathways by which alcohol has to get metabolized. Alcohol is the only chemical constituent known to the human being which has to get necessarily metabolized. This is the only thing which just simply cannot be inertly stored. So as soon as alcohol reaches the liver, it has to get metabolized. There are three ways by which it gets dehydrogenated. The first is that then the most important mechanism of its oxygenation or dehydrogenation is that it is acted upon by an enzyme called aldehyde dehydrogenase, which takes a hydrogen from which gives a hydrogen from NAD to NADPH, leading to the production of acetaldehyde. In case this mechanism gets exhausted, you have stimulation of the endoplasmic related mechanism, which is also called cytochrome P450 mechanism, by which you lead to production of NADPH from NADP. And finally, with these two pathways get exhausted, you have accessory pathway called production of the peroxisome, in which H2O2 is converted into H2S. Once the major metabolic pathway has taken care of by converting alcohol into acetaldehyde, acetaldehyde quickly moves into the mitochondria, where another enzyme called acetaldehyde dehydrogenase again takes the hydrogen away from it, leading to the production of NAD and the depletion of NAD, resulting in an altered ratio between NAD and NADH, leading to the production of the acetate. Once alcohol has been converted into acetate, you can sit pretty about it. This acetate then moves into the circulation and is utilized by all the organs as a food material. So what is this alcohol metabolism resulting into? It results into two important components. As the alcohol is getting converted into acetaldehyde, not all acetaldehyde will subsequently be converted into acetone. So a number of acetaldehyde and its products, they tend to get accumulated in the liver as well as into the circulation, resulting in this so-called acetaldehyde adducts. This acetaldehyde adducts, which is the melanoidaldehyde acetyl complex, this gets accumulated in the liver in patients who continue to drink alcohol. In addition, because of the consumption of NAD, you have a reduction of NAD and more NADPH. So this leads to alteration in the so-called cellular redox potential, or alteration in the ratio between NAD and NADPH. So what alcohol is resulting is in two things. You have accumulation of acetaldehyde and its product, as well as resulting in oxidative stress, which is so important when we come to the injury caused to the liver by alcohol. So anything which you drink, especially if this is a standard drink called 14 gram of alcohol, you can see it by the different ways by which you consume alcohol. So anybody who drinks alcohol, 90% of those who are drinking more than 4 drinks will develop alcoholic fatty liver as quickly as 2 weeks after starting the consumption of alcohol. So if you stop drinking alcohol, it is absolutely reversible. But those who continue to imbibe alcohol, they have two fates. 10 to 15% of them will have triggering of the inflammation occurring because of the presence of the fat in the liver. And of those with inflammation in the liver called steatohepatitis, about 1/3 to 50% will develop fibrosis of the liver or cirrhosis of the liver. So you have a normal liver which is acted upon by alcohol, resulting in fatty liver, which at some stage of life, we still do not know why there is a shift over of the gears from fatty liver to steatohepatitis and from steatohepatitis to frank cirrhosis of the liver. Let's start with the basic pathogenesis as to why we develop fat in the liver once you start consuming alcohol. Two important factors are that if you start drinking alcohol, that is about 4 to 5 standard drink. A standard drink I've already mentioned is around 14 gram of alcohol, absolute alcohol per day. And within few hours, you will develop fatty liver, especially if you're drinking in the form of a so-called binge drinking. Now, binge drinking is defined by somebody who continues to drink 4 to 5 drinks in a period of 2 hours or less. What happens with consumption of alcohol is there are three ways by which there is a fat accumulation. First is alcohol per se accelerates hepatic lipogenesis. That means you are producing a lot of fat in the liver. It decelerates or decreases the hepatic lipid breakdown. That means whatever fat is present in the liver, it will not be broken down further. And subsequently, it also decreases the exit or the export of the fat from the liver. All of this result in the formation of fatty liver disease. Starting with the deposition of the fat in the liver. Once you have ethanol or acetaldehyde oxidation, it leads to generation of the lot of NADH from NAD, which I have already shown. This results in alteration in the cellular redox potential, which leads to enhanced lipogenesis or lipogenesis. So what happens is the acetaldehyde and its products, they lead to the migration of this protein called sterol regulatory elemental binding protein, or SRAP1C. So this moves away from endoplasmic reticulum onto the Golgi apparatus. This is facilitated by the altered redox potential, which has happened because of the persistent utilization of the alcohol in its attempt to metabolize it to acetaldehyde. Now, once this SRAP enters the glucose, it enters the Golgi apparatus, it gets matured, it gets transcriptionally active, and leads to production of the supranuclear factor in the form of SRAP protein. Once it goes into the nucleus, it leads to stimulation of a number of lipogenic enzymes, which increasing expression of these enzymes. And I'm sure all of you are familiar with number of these enzymes which are responsible for production of the fatty acid, not only from the lipid, not only from the fatty acid, but also from the glucose and proteins by using this different set of enzymes, which are called lipogenic enzyme. Another thing which is happening is at the same time is there is an overexpression of this supranuclear factor called early growth response 1 or ERG1. Now, ERG1 controls the expression of the genes that respond to cellular stress. It binds to the gene promoter region that are relevant to alcohol-induced liver injury and steatosis. So two things are happening. You are having SRAP stimulation, which leads to stimulation of the lipogenic enzyme, and then you have stimulation of ERG1, which leads to stimulation of lipogenic cytokines, which tend to get stimulated because of this nuclear factor going and stimulated the genes, of which the most important is TNF alpha. So you have an alcohol which is getting in the cytoplasm metabolized to acetaldehyde. Acetaldehyde moves into the mitochondria, where it leads to stimulation of EGR1 mRNA, which is the dual mechanism of increasing the TNF alpha, which is not only important lipogenic stimulant, but also increases the expression of the SRAP1C, which is important for stimulation of the lipogenic enzymes. Second thing which is happening in patients with alcoholic liver disease is that in a normal course of the events, if you have lipid which is stored in the liver's hepatocytes, it is picked up by the autophagosome, and autophagosomes make it fuse to the lysosomes, where on the lysosomes you have lipases which try to break down into the free fatty acid, which are typically taken care of by the mitochondria, leading to beta oxidation. But in alcoholic patients, this autophagosome is restricted. So that means whatever fat is getting accumulated, there is no way it is being converted into free fatty acid for beta oxidation in the mitochondria. So you have retarded autophagy, you have less and defective lysosome, and resulting in a reduced beta oxidation. So that means whatever fat is present, it is not being oxidized in the mitochondria, resulting in the accumulation of the fat. In fact, there are other mechanisms of reduced beta oxidation in patients with alcoholic liver disease because of the altered redox potential, which I have shown you, because of the altered ratio in NADPH ratio. You have mitochondrial instability, so the beta oxidation cannot occur. You have reduced production of the adiponectin from the fat cells, as well as the inactivation of the PPR alpha, which is important for the migration of the fatty acid into the mitochondria for its oxidation. All this results in the reduced beta oxidation. Third thing which is happening in patients with alcoholic liver disease who tend to develop fatty liver is that there is a defective export of the liver. That means even if the liver has got fat, in the normal course of the events, it is combined with cholesterol and is extruded out as VLDL, that is very low density lipoproteins. Something happens in patients with alcoholic liver disease, that there is impaired synthesis of this, and there is impaired export of the fatty for VLDL from the liver out into the circulation, resulting in the accumulation of the fat in the liver. So in essence, if you have been drinking regularly, enhanced lipogenesis, arrested lipophagy, decreased adipokine production, impaired excretion of the VLDL out from the liver, as well as the increased influx of the fatty acid from the adipose tissue, all culminates in the formation of fatty liver disease. Now coming on to what switches the gear from fatty liver to steatohepatitis. That is important. All of us have seen patients who have been just going around with fatty liver disease. What happens suddenly that these patients come to us with alcoholic hepatitis? Two important factors which change the patient's fatty liver into steatohepatitis include, again, acetaldehyde adducts, which have been constantly being formed in patients who continue to imbibe alcohol, and the migration of lipopolysaccharides that is resulting because of the endotoxemia from the leaky gut. So these two mechanisms, they tend to stimulate the sleeping dog called Cooper cells, and they tend to become active. So a combination of acetaldehyde adducts, which are coming from the liver, because there is a rate limiting step of converting acetaldehyde into acetate in the mitochondria. So a large number of acetaldehyde adducts coupled with endotoxin coming from the GI tract because of the permeable intestinal membrane, results in the activation of the Cooper cells, which are normal residents and part of the liver parenchyma. So the resident macrophages, which are important for maintaining the immune status of the liver because of the incoming pathogens from the GI tract in the normal course of the events, they tend to attract the infiltrating macrophages. So the Cooper cells and the cells which are migrating from the circulation, they tend to land up in the liver producing inflammation. Normally, Cooper cells are the resident liver cells which give you the first line of defense against the incoming pathogens or PAMs from the GI tract. Infiltrating macrophages will be recruited once there is a stimulus or the cytokines which attract the inflammation towards the liver. So in the normal course of the event, if you have a bit of inflammation coming from the GI tract, so you will have a combination of the pro-inflammatory macrophages called M1, which will be stimulated, and partly then subsequently it will be neutralized by M2 type of cells, which give you an anti-inflammatory stance. So what determines whether the sleeping macrophage will get converted into pro-inflammatory cell or into anti-inflammatory cell called M2 will depend on what type of growth factor, cytokines, PAMs or DAMs you have, which are responsible for a switchover of macrophages from pro-inflammatory M1 to M2 and vice versa. But if you have alcohol, you have substantial shift over and polarization of the macrophages into the pro-inflammatory or M1 type of phenotype. And this, once you have stimulation of the pro-inflammatory M1 macrophages, so which it is a very rich source of reactive oxygen species which are detrimental for the liver, and it also is important for attraction of the inflammatory cells down from the circulation. So what triggers patient's activity in a patient with a bland liver is if you continue to drink alcohol, you have a GI tract which is acting in conjunction. So GI tract's increased intestinal mobility leads to the endotoxemia, which is picked up the LPS from the GI tract coming through the permeable GI tract is picked up by the TRL receptors and the Cooper cells, leading to the stimulation of the pro-inflammatory cytokines, which release a number of cytokines resulting in inflammation and injury to the liver called steatohepatitis or alcoholic hepatitis. So this is what is happening. You have alcohol which is directly damaging the liver. You have the intestine which is getting affected from the intestines. So large number of pathogen-associated molecular pattern coming from the GI tract as well as the injury occurring within the liver parenchyma because of the direct effect of alcohol resulting in apoptosis. A combination of the PAMs and DAMs on converting these resident Cooper cells, or which are called resident macrophages, into stimulated or M1 type of pro-inflammatory cytokines, which produce a number of pro-inflammatory cytokines, which lead to perpetuation of the injuries, resulting in changeover from a bland fatty liver to the occurrence of alcoholic hepatitis. Gut is a big contributor to this. I think Dr. Sujit has already mentioned a lot about it. So a combination of the bacterial overgrowth, bacterial dysbiosis, increasing intestinal hyperpermeability, as well as the modulation of the mucosal immunity at the level of the gut is important for changeover of steatohep from fatty liver disease to alcoholic hepatitis. And we know very clearly now that the good bacteria in the form of lactobacillus and bifidobacterium, they tend to decrease, and you have increased concentration of the protobacteria and enterobacteria in the GI tract. Gut-derived inflammation.
Sir, may I request you to conclude, please?
Yeah. So the gut-derived inflammation he has already talked about. It is disrupted by patients who have been consuming alcohol, resulting in increased hyperpermeability, which he has already talked about. And finally, you have patients who continue to imbibe alcohol. At some point of time, these Cooper cells end up stimulating hepatic stem cells, which release pro-inflammatory cytokines, which result in development of the fibrosis. So at the end of the day, you have inflammation in the liver, which was triggered by stimulated Cooper cells, finally leads to stimulation of the hepatic stellate cells, resulting in production of the fibrosis. So ladies and gentlemen, you have fibrosis and cirrhosis, which tends to occur in the late stages of alcoholic liver disease because of the deposition of the fat in the space of the disc. To sum up, there is an initiation of the injury followed by perpetuation of the injury because of the continuing imbibition of the alcohol. And to sum up, ladies and gentlemen, major burden on the liver is to oxidize the alcohol as soon as it reaches the body. And it is an effort to remove the alcohol from the body. This results in production of acetaldehyde and its adducts. This, coupled with endotoxemia, result in production of the fatty liver disease, which is further compounded by alteration in the gut mechanism, resulting in the endotoxemia and aldehyde products, which gives you a range of problems within the liver, starting from the fatty liver, then triggering of the inflammation, finally resulting in cirrhosis of the liver. Thank you. I'll stop sharing my slides.
Thank you, Anil. I think you really went into the area, but I'll be pardoned by you and Dharmesh, because what I looked at is macroscopic alcohol injury and a microscopic alcohol injury. When you take hepatocyte itself, leave the stellate and Kupffer cells by side, I think it has a different injury. What I learned and understood is it's more of a mitochondrial injury, where we even say that the GGTP elevation is because of mitochondrial injury. I know Dr. Muthuswamy Hariharan and Dr. Manisha, they will comment after Dr. Dharmesh comment. I expect, both of you are tigers, and it appears as if I gave the same drink to the same tigers. So I'm sure Dharmesh will twist the whole thing and play the game very well. Welcome, Dharmesh.
Thank you so much, sir, and good evening to all the colleagues and friends and fellows in training who've logged in for this wonderful syllabus course. So the brief given to me is toxic effect of alcohol on hepatocytes. As always, sir tries to hit below the belt. So the first thing to say is that the hepatocytes per se cannot produce the kind of significant injury which has been described by Dr. Anil in the earlier talk. So we all know and that alcohol is a serious problem, and in the interest of time, I'll skip whatever Dr. Anil has already shown. And what just intrigues me is that at least in some parts, the stages of alcohol-related injury to the liver are reversible. So again, that is something that we don't talk about oftentimes. So even beyond the steatosis stage, we know that there are people who present even with acute on chronic liver failure due to severe alcohol, but they also have significant reversal of injury. Unlike the acute liver failure scenario, we still do not know what happens to the histology in these patients. So is this completely reversed to normalcy, or is there still some lingering effects that are seen? My own feeling is that there will be still some lingering effects seen, but again, this is something which has not been researched extensively or published in peer-reviewed literature. Now, this is something that Dr. Anil alluded to a little while ago. And in fact, just with this one slide, you can complete the topic that has been given to me. So the bulk of the alcohol gets metabolized by the liver by the alcohol dehydrogenase, or the ADH. So please remember not to be mistaken for antidiuretic hormone.
Now, do you have a slide? I'm sorry, Dharmesh. Do you have a slide?
Sorry?
Do you have a slide to share?
Sir, is it not visible?
No, not visible. No, you didn't share it. Please.
Oh, I shared it right in the outset, sir. Oh my God.
Sir, because that
it is not shared, Dharmesh.
Oh, it's very strange.
No. I think you just try it. It will come.
Jordan, please help.
Yes.
Jordan,
can you just open your Zoom window and click the share screen button. You'll have to open Zoom, click the share screen button, and
Yeah.
click your presentation there and share.
Can you see it?
No,
No,
not.
not.
But I clicked on the share screen.
Dharmesh sir, first do the stop sharing and then again do resharing. You must have played it side.
Okay,
Yes.
can I?
Yeah,
Can
it's
you
coming.
see it
It
now?
is coming.
Yes,
It is coming. Yeah, you got it. Yes.
Sure.
Yes. You can
Sure.
move. You have
I'm very
21.
sorry for that.
No,
So
no
this is-
problem.
Yeah, this
No
is the
problem.
slide that actually needs attention, and